afternoon, everybody. My name is Sasha Merrill, and thank you so much for joining me. I am currently an incoming plant science major. I am transferring from San Diego Mesa College, and today we are going to be talking about preventing insect egg death due to fungal overgrowth. This summer, I was lucky enough to be able to work in the joined lab of Dr. Peter Atkinson and Dr. Linda Walling. And one of their main goals right now is actually studying the genomes of specific agricultural pests. And the ultimate goal is that if we can learn about these genetics, we can then kind of use them to our benefit and reduce the effectiveness of some of these crop pests. The problem is the species that we are currently studying, their genomes haven't really been studied very much. So what we have to do is take the egg masses and we set them on agar plates. They're then micro-injected with various CRISPR-Cas9 components, and then you have to wait typically about a week, sometimes a little more, sometimes a little less, for the insects to actually hatch. And then once you have the nymphs, you can kind of see what they're doing. So there's a lot of really new and innovative things that are going on in the lab. And so after you lay your plates, you have to go and check on them every single day and see how the nymphs are doing and make sure everything's growing. And also to get rid of any fungus that's growing, because the agar plates are set with mesh screens over the top so that the leaf and the eggs can get airflow and moisture because they're kind of sitting on top of a water bath. Well, the fungus gets everywhere, and that can tend to be a little bit of a problem. It can affect the leaf itself, preventing the eggs from being able to be viable enough to hatch. The fungus can grow on the eggs themselves, and it can be a huge waste of time to have to go through and clean this fungus every single day, in addition to all of the Q-tips that we're wasting, and then perhaps the material that we've been injecting. Besides that, it can also just act as this extra random variable when we're looking at our data. So it's kind of hard to tell, did the egg die and then the fungus took over, or was it an error that was done by the microinjector, or was it perhaps the CRISPR-Cas9 that was put in that did it? So my ultimate goal is to try to kind of whittle down some of these excess variables. And then the picture here actually shows the agar plate, and then the little smiley face looking thing is going to be the egg mass. There's actually going to be several eggs hanging out in there, and then they have to be individually micro-injected. So essentially, what I did is I collected the egg masses as we normally would, and then I cleaned them with different techniques. So one was just rinsed with deionized water, another one was soaked in bleach, I soaked one in ethanol, and then another one in a Benamil and water mixture. And then we had to go through and I micro-injected the eggs. For this experiment, we just used sterile water because I just wanted to make sure that the eggs could survive the cleaning process and being injected themselves, because sometimes when they hatch as nymphs, you can actually see the scars on them from where they've been micro-injected. So it can actually affect the eggs. So you want to make sure that they can handle everything that's going on. And then I go through and I would check the leaves every single day and clean off my fungus and see how they were reacting to the different treatments. And so the two pictures here are just going to kind of show what the plates look like before they have been prepped, and then afterwards when they've been micro-injected, and then those are the guys that I kind of have to keep an eye on. So for our first trial, I noticed that the first day pretty much always goes really well. There's not normally too much going on. Everything's still kind of getting adjusted to its new environment. We had actually expected the bleach to last the longest because that was the cleaning method that we've been using in the lab currently. And out of all four of the plates, that was the only one that I had to throw away because it just got completely overrun. And then it was really interesting because the Benamil mixture, it didn't have any fungus present, but there was some bacterial growth because the glassy wing sharpshooter, that's one of their main disease vectors, is a bacteria that they transmit that kills the plants. So you do occasionally see it on there. It's just not as much of a problem as the fungus can be. So that kind of led me to a little midway point. And so the top picture, the red circle, is actually going to indicate where my eggs were at one point. And the fungal growth actually started on the leaf right above the eggs. And so it just kind of, part of it is still always going to be a random variable, but we did notice that the ethanol and the benamyl were typically more consistent. This experiment actually had been run by one of the grad students in my lab prior to me coming in, and his experiment didn't really yield too many like tangible results. And so he was like, well, let's have you try it and see what happens. And mine kind of did the same thing. So we kind of decided that but since the ethanol and the benamyl regularly did better than the other ones, that perhaps we should try to mix them together and then see how the leaf would react to that. And then also, make sure that it won't bother the nymphs themselves too much. So for that one, we did 5 trials. It was the 40% ethanol mixed with 2,000 parts per million of benamyl. And out of the 5 trials, we actually only had 1 plate that had to be completely discarded. The rest of them had very high survivability rates. So that was actually pretty exciting. The 2 plates on the right is actually going to be, on the top is day 5, and then on the bottom is day 6 of the plate that wasn't able to survive. And we did notice that up until about day 4, everything stayed pretty green. They looked almost exactly the same. And then once, it's been more than a few days, they start to rapidly deteriorate, and it kind of becomes a little bit of a race against the clock. But this race we did pretty well in. So that was fairly exciting. And ultimately, we kind of just realized that there's always going to be some random variable in science, especially with fungus. You never know where it's going to decide to pop up and grow. And also the hatching rates of the glassy wing sharpshooters themselves do tend to fluctuate quite a bit for insects. So that's another thing that we always kind of keep in mind. But ultimately, the benamyl ethanol mixture was really effective. It didn't hurt the eggs, and it did a really good job of keeping everything kind of put together for long enough, I should say. And perhaps in the future, we could see if there are different concentrations of the ethanol or the benamyl itself, or perhaps mixing them in a different ratio might be kind of beneficial. But it was really interesting doing this project because I have been able to work in all parts of the lab. So I've been able to do some really cool stuff with like the CRISPR and the Cas9 and seeing how they do all that research. And so I was kind of joking, I'm like, my presentation's about fungus though. And so it might seem a bit lackluster, but like everyone in the lab was so excited about my experiment. They like, oh, it went so well. Like, we're so happy about this. And it kind of like reiterated that fact to me that every little bit of science is always going to be helpful. Like, even the little things, and it just gives us something else to grow off of. So that was kind of like a really cool little thing that I didn't expect to learn from this. And finally, I would like to thank everyone for sitting here and listening to me talk about fungus for 10 minutes. And then also, I spent so much time ping-ponging all over campus, out in ag ops and in the insectary and the genomics building and in entomology. So thank you to everyone who let me in everywhere and who taught me how to do all the different things. I had so much fun this summer. Thank you to all the programs, to Riverside for allowing me to come to the school at all. And I'm definitely looking forward to learning more stuff and actually starting classes in the future. And if there's any questions, I would be more than happy to answer them. Thank you.